Good morning, everybody. I'm Sandy Boyce. I'm the conference chair, and it's a pleasure today to introduce you to the plenary speaker. Before I do that, just a couple words here. Research is a broad topic, but applied research is really important. And the reason is they're real world problems that people can tackle and find solutions to. Dr. Wilde is one of those people who actually takes on some of those really nasty problems and solves them. So you're really going to enjoy her talk. Everybody in here who publishes would like to publish in Science. It's a top-notch journal. And Dr. Wilde not only published a great article in Science, but she also won the Best Paper of the Year award for her work. Astounding. So it's just a really great achievement for her. And everybody else as well, because we're all interested in raptors. And of course, she took her place in the long list of people who care for raptors. She's a professor, associate professor at the University of Georgia. Her interests are in aquatic systems. And this is an amazing story, and I think you're going to love it.
Thanks so much. I'm really excited to be part of this conference. I haven't been here before, but I've met a lot of wonderful people and I've learned a lot already. So I have sort of a gruesome story to tell, but I think an important one. And in the realm of all the work that you're doing, I hope that you'll talk to me about it. So I've been doing this for a while, and certainly not on my own. There's a whole host of people that have contributed to this research over the last 20 years. And so we'll end my talk with acknowledging them, but we'll start off way back in Arkansas when these eagles first started dying from a novel disease and presented with vacuoles in the myelin sheath of the brain. And there was no explanation for it. And it seemed like the research funds kind of went away, but I was very obsessed with it. So starting in 2001, when I was at the Charleston lab, I started working on this. And we've been working through a lot of different monitoring studies and food chains, studies to be able to determine what the source of this mortality was. And coming to the end, now we're concerned about human health. So yes, that was amazing. We had a whole host of many other authors, but we had two students that got the first author, co-author, and then Timo Niedermeyer and I were the primary senior authors, but many more authors on that paper. I want to see if this will run because it explains a lot of what's happened so far.
The AAAS Newcomb Cleveland Prize, our oldest award, recognizes an outstanding original research paper published in Science each year. The winning paper is chosen based on the quality of its scholarship, innovation, presentation, likelihood of influencing its field, and its wider interdisciplinary significance. This year, we present the prize to the paper entitled Hunting the Ego Killer, a cyanobacterial neurotoxin causes vacuolar myelinopathy, first published March 26, 2021. The paper was the product of an international collaboration co-led by Susan Wilde, Associate Professor of Aquatic Science at the University of Georgia, and Timo Niedermeyer, Professor of Pharmacognosy at Martin Luther University, Halle-Wittenberg in Germany. At DeGray Lake, Arkansas, in fall 1994, a mass die-off of bald eagles initiated a wildlife investigation. Dozens of birds died, and many displayed odd behavior, like missing their perches and flying into rock walls. When wildlife biologists examined dead eagles, they found extensive lesions throughout their brains and spinal cords. By 1998, scientists knew that birds from at least 10 different southeastern reservoirs in 5 states had this new disease called vacuolar myelinopathy, or VM, but no one knew the cause. In addition to avian species, Susan Wilde and UGA colleagues documented that VM could affect amphibians, reptiles, and fish. Researchers made gradual progress uncovering the cause. Wilde and her colleagues in South Carolina, where many of the birds suffered from VM, found dense growth of the invasive plant Hydrilla verticillata. The plant, imported from India for use in aquariums, was overrunning man-made lakes in the southeast, readily consumed by coots, which are frequently eaten by bald eagles. Using fluorescence microscopy, Wilde observed that in affected water bodies, hydrilla leaves were densely covered with cyanobacteria colonies. This novel cyanobacterium was also found in all the reservoirs where the birds had died. Wilde and her colleagues speculated that the cyanobacterium produced a toxin responsible for the eagle deaths. They fed plants collected from affected water bodies to laboratory birds, confirming the plants colonized by cyanobacteria caused VM. The cyanobacterium was named Autochtonos hydrolicola, Greek for eagle killer living on hydrilla. However, no known toxin was detected in the cyanobacterium. In Germany, Niedermeyer and co-authors grew this cyanobacterium in the lab. However, lab-cultured bacteria did not cause VM in test chickens. Hypothesizing that the toxin might be produced in lakes but not in the lab, they collected more hydrilla leaves from impacted lakes. Direct analysis of the cyanobacteria growing on these leaves revealed a compound not present in lab-grown autochtonos that contained 5 bromine atoms. The team was able to reproduce the compound in the lab when the cyanobacteria were grown in the presence of bromide. The compound was revealed to be a novel biindole alkaloid, and further experiments confirmed it causes VM. The scientists called the compound a toxin that kills the eagle. Jana Marish at the Biology Center of the Czech Academy of Sciences led the genome sequencing of Autochtonos hydrolicola, which allowed the identification of the compound's biosynthetic gene cluster. Bromide can leach from rocks, but it can also be introduced into the environment from human activities such as coal-fueled power plants or water treatment facilities. The authors recommended increasing monitoring and public awareness campaigns be implemented for hydrilla and autochtonotoxin to protect both wildlife and human health. The study brought researchers together from multiple countries and disciplines to solve a decades-old mystery and reminds us how dangerous human-induced changes to ecosystems can be. These unanticipated effects are only likely to increase as we continue to alter the world around us. Hunting the eagle killer, a cyanobacterial neurotoxin, causes vacuolar myelinopathy, recipient of this year's AAAS Newcomb Cleveland Prize.
We have been working on this for a while, and initially, of course, it was noted in eagles. They're a pretty dramatic bird. The public tends to recognize them and think it's important and turn them in. But we also found that the great horned owl territories in these same reservoirs were failing. They're nesting there year round, so very vulnerable. The bald eagles come down in August and they're setting up territories, and then they have their young in the spring. So they're there during the time when the toxin is produced by the cyanobacteria. But we have lots of other herbivorous waterfowl that are also affected by this disease, and they come in migration hungry to our hydrilla infested reservoirs and eat a lot of this hydrilla and start flopping upside down. So that was the most direct link to go from our invasive aquatic plant with the toxic cyanobacteria to a very abundant herbivorous migratory waterfowl, and then into our bird of prey. But we have noted it in some other birds, like this little killdeer. Just going to tell a quick story about this hunter. So this Canada geese that he was holding he had harvested it from an AVM reservoir in South Carolina. So I asked if I could have the heads for the histology, and he said, sure, but I want you to take my picture first. But we found marked lesions in that goose's brain. So I've always been a little worried about hunter and fishermen. So pretty slow at this research. It took me a really long time from the time when I first saw it in 2001, and I said, aha, this must be it. It's toxic. It's a cyanobacteria. In the freshwater realm, they're the ones that make the most dangerous toxins. And I had never seen it before, and I couldn't find it in any of the taxonomy guides. And it was very abundant, but only on the underside of the hydrilla leaf. But because it was a brand new species and genus, and they weren't even sure what family to put it in, you think birds are complicated taxonomically. Try on cyanobacteria. There's people that publish every day a new species or just change them. But in any case, we did publish this, and it's Autochtonos hydrilicola. They kind of said it wrong in the science video. You guys mind? Autochthonous hydrilla. Oh, okay, I'm not gonna say that. And it's really cryptic on the leaves if you're looking at it under the microscope. But you can see these spots on the leaves when there's a very dense colony. So that would be like 100,000 cells on that leaf. But because it's green on green, I was using fluorescence microscopy to be able to actually see it, which I don't know if you can or not, but this allows us to, with top lighting, then to just see the autofluorescence of the cyanobacteria because only their pigments show up. So you can actually see that this is the most, this coverage across the leaf is pretty extensive. And we have seen hydrilla expanding up into the Northeast and across the United States, that so far I've only found the cyanobacteria growing from Texas to Virginia and Florida. And we have tested a number of different taxa with feeding trials. It's sort of cruel, but in order to be able to determine whether or not they're susceptible to this toxin, we had to actually do feeding trials. And I'll go through a few of these that we have done. And I would say we, because a lot of this was senior thesis students and graduate students. But the turtles, the herbivorous turtles that were consuming hydrilla directly in our trials didn't display clinical signs for almost 3 months, but then they started to have this ataxia and weakness, lethargy, and they presented with the same vacuolar myelinopathy in the lesions in the myelin sheath. We also did some feeding trials with tadpoles that fed on the hydrilla that had the cyanobacteria growing on, and hydrilla that as a control that did not have this cyanobacteria growing on it. And within 30 days, we lost our tadpoles that were consuming it directly. Interestingly, so far, the only thing we've tested that doesn't seem to be affected was a green tree frog. So I don't know what to do about that. And this was a second set of experiments where we're basically looking at trophic transfer of the toxin. So we had to gut load the prey with the cyanobacteria, hydrilla. So the snails and the tadpoles ate it directly, and then we fed them to these pedomorphic salamanders. They have an aquatic stage. So they were quite vulnerable to it and couldn't even right themselves. I'm going to mention seizures here because that has been a consistent clinical presentation of this disease, and don't know yet which precedes. If you have vacuolar myelinopathy, if you get lesions in your brains, you might cause seizures. If you have seizures, it will cause lesions in your brain. So we don't know which is which yet. And with the second trophic transfer experiment we did was with the herbivorous fish that were fed to the water snakes, and they didn't appear to really change behavior very much. We did have a few that were so anorexic we had to euthanize them before the end of the trial, but they had the vacuolar lesions in their spinal cord, especially in the cervical region. And I stuck these slides in here because we're in Florida. One of the former invasive plant managers here in Florida said, what if the snail kites could get it by eating these invasive apple snails that are eating hydrilla? And I said, I don't even think you'll have it in Florida. Like, this is in the Piedmont across, the Southeast. I doubt it would be there, but we started up in top of the Semi-Chain Lakes and drove all the way down through the canals and collected the hydrilla the whole way. And I did find it in Lake Toho that first time that we sampled more extensively. And so we started collecting some apple snails from locations where we had found the cyanobacteria growing on the hydrilla. And so we're not going to do experiments on threatened and endangered species. So we are just feeding this material to chickens as our bioassay to determine whether or not the toxin was present. We had to do all of these bioassays before we knew what the compound was, because we couldn't determine the concentration of an unknown toxin, right? So we just had to see what effect it had on the organism. And it would cause the chickens to not be able to stand up anymore. They had a lot of hind limb ataxia, and they developed lesions from consuming either the hydrilla or these big invasive apple snails. that love to eat a lot of hydrilla. So sort of prove the concept that the snail kites are likely getting some of the toxin when they're consuming these exotic apple snails. So we're going to work some more with Rob Fletcher to determine like exactly what portion of that apple snail the snail kites are consuming, and look at pieces and parts and see where the toxin's actually located within those organisms. And if we recover snail kites, they don't have to be fresh enough for histology anymore, because now we know what the toxin is and we can use analytical methods to determine it. And unfortunately, it's quite stable. We have been working at J. Strom Thurmond or Clarks Hill, if you live in Georgia, for a long time, and working with the Army Corps of Engineers to try to deal with the situation there where they had a lot of hydrilla and a lot of dead bald eagles over many years. And they have stopped grass carp now, and this situation is better. But we do feel like that when the migratory birds are coming down and selecting these reservoirs, and then the eagles are very much, they're moving to these reservoirs because there's ample prey there. So we're kind of bringing them into a habitat that is not safe, but it looks really good. And I just had to put a picture of Libby in here working. She came down to help us do a very tiny satellite telemetry project. She was like, you guys don't have enough money to do satellite telemetry. We're like, we want to do a couple birds. So she came, and my graduate student, Bridget Harm, also had a lot of experience that really made a difference. But we were able to document that if you were hatched out of J. Strom Thurmond nest, you were gonna, if you survived that season, you come back, and we saw them mortality at J. Strom Thurmond, the eaglets that were successful in hatching that year. So moving on to the next part, this is really more of the discovery phase for the analytical work. And a lot of this work was done by Timo Niedermeyer, and he is the expert on natural products chemist. So I'll just tell you what I know about it. I mentioned that we have these bioassays. Initially, we were using this cell culture, which would correctly tell us every time that the cyanobacteria was present. It was a good determinant for that, but the cyanobacteria doesn't make toxin all of the time. As they alluded to in that video, there's a window of time in the field when we see the toxin produced. And in the laboratory, it was only when we added bromide to the cultures that we saw them be able to produce that toxin. So for that, we had a bioassay. The invertebrates are these little Seriadaphnea dubia, and then zebrafish are actually a really powerful model for human work, because we share so much of their genome, and they're really tiny and they reproduce rapidly. So that allowed us then to just go to that peak on the HPLC trace and collect only that peak and test it on our bioassay organisms. And now that's something that you can take and go to NMR and try to do some more discovery work on what that molecule looks like. Because as you can imagine, the hydrilla plus the cyanobacteria that we collect from the field has so many other things in it that once we extract it, and it looks green and gooey and nasty, and nobody wants that on their HPLC or anything else. So we had to clean it up without losing the toxin. And this is a zebrafish. Let's see if they'll start swimming. Slide from my student Tabby Phillips, and she's been working with James Lauderdale, a neuroscientist at UGA, looking at how they're affected. And this is just little 5 to 7 days past fertilization zebrafish, and they're clear. So you can see their little heart and their little pectoral fins. They're trying very hard to swim in the AVM treatment, whereas the normal solvent control, they're looking pretty normal, zipping around. So she did a series of dose response curves for the zebrafish, and it appears pretty consistently toxic at a fairly low level to the zebrafish when they're in a water bath exposure to that. And she did a kind of cool little thing where she drew how far they were swimming, whether they were in a control versus the treatment, and they kind of stay in the same place. They're sort of stuck once they get dosed with the toxin. And the other interesting finding is that they have really severe seizures, and those seizures are long, like elongated seizures that had been observed previously. So we know that there is something that's initiating that seizure response, as well as these lesions. And I can tell you more detail about this. It's a way of visualizing calcium channels that turn on whenever the toxin is presented to the zebrafish. So it could actually be something that's messing with calcium channels in the cells. And this is really a sad video, but it shows you what our sentinel mallard looks like after he's been out in a lake eating hydrilla. And this, of course, is before we still had to use these bioassays to tell if it was going to be bad. So he's starting to have the tremors and a seizure. And of course, when they present like this, we will euthanize them in the field. So the neat thing about working with this amazing chemist is we have a lot of new tools that they can use. And this MALDI MS actually allows you to look at the hydrilla leaf and not only see where the cyanobacteria colonies are on the surface, but also localize where that toxin is on the surface. So then the second panel there pops out that brominated compound. It's going to show that all the different bromines, plus it could either be 5, 4, 3, depending on whether or not it's been broken down or not. And we found that the source of the bromide in these systems is probably the hydrilla itself. It's hyperaccumulates compounds. A lot of submerged aquatic vegetation can hyperaccumulate some pretty dangerous compounds, but that's a good thing for bioremediation. But if your ducks are eating it, it's a bad thing. So these hydrilla was able to actually sort of concentrate the amount of bromide that is in the stems and in the leaves, and that's where my cyanobacteria is growing. We also found really high levels in Lake Okeechobee, but I've never found the cyanobacteria growing there. There seem to be a couple of things that define whether or not the cyanobacteria is going to grow there if hydrilla is there. And one of them is fairly low conductivity. So I don't think the springs are going to be in danger. And the other is a low pH and less productive, say, sediment, more of a granite base, which is what we see in the Piedmont. But here in Florida, where I've seen it so far, it's just east of the Florida Ridge. So a little bit different landscape there. I'm going back and forth, aren't I? So this was Stefan came over to the US and worked with some of my students on the HPLC, and he was working with Bridget Harum, and the two of them are looking at trace, and he was like, well, what about that big one at the end? She goes, oh, I think that's just pigment. And he's like, no, it's not in my cultures, but you know, it's in those ones from J. Strom Thurman. So together they really started figuring it out because they both had pieces of the puzzle they put together. So anyway, once he added the bromine to his culture, he started to see production of that peak of the toxin. And it is a pretty cool molecule. All those bromines on the outside, the nitrile groups. It appears to be quite toxic. It is lipid soluble, which might have been one of the reasons it was so hard for us to describe it to begin with, and stable under heating or freezing. So it's going to be around for a while once it's formed. And Tabby was able to get to do the electron microscopy as well as the light microscopy to visualize the lesions, both in just the field collected hydrilla autochthonous mixture and the purified toxin. She was very excited about the oligodendrocytes exploding because this type of vascular myelonopathy is caused by a splitting of the myelin sheaths. So it's a swelling at that node. So that is how you can actually definitively say that it has intramyelinic edema. And we did find the toxin in fish fillets that people eat, as well as the plants and their gut tracts. So this was some fish that were collected from J-Strom Thurman again, and there was a significant amount of the toxin in those fish fillets of game fish. And the first time we submitted our paper to science, they said, okay, fine, but you got to bring it all the way back around to the coots. We want to see whether or not the coots have the toxin in their tissues. And we did have some in our freezer. I have lots of stuff in my freezer. So we tested it now that we had the method, and we did find pretty significant levels in the breast tissue and thigh tissue. So it seemed like the fattier tissues were a little higher. What are we going to do about it? I have to drink water when I'm nervous. Sorry. Doing great. So there's a number of different ways that people try to control hydrilla. It's kind of funny that we think we can do that. And the only one that I've seen really be truly successful is a little scary to introduce the biological control, the sterile grass carp. Because once they run out of hydrilla, they'll just be eating everything else you have. But our southeastern reservoirs aren't real systems. We don't have native aquatic plants most of the time. And I would argue that if this aquatic plant is dangerous to fish and wildlife and probably us, then we don't want it in there. So treating it with chemicals is more likely to release that bromide and cause a premature senescence of those hydrilla plants. And then they'll produce toxin earlier in the year. And we've done that experiment with the Army Corps of Engineers a couple of times, because we went out to collect early season hydrilla. We expected it to not be producing toxin. And they're like, oh, we treated with diquat dibromide last week. So, it was toxic. So I feel like that's probably what happens in Florida, since you don't have that seasonality that we have in the Piedmont. If you are killing the hydrilla back with hurricanes or a quick cold snap or chemicals, then it would be able to release the extra nutrition and that bromide that the cyanobacteria needs to make the toxin. So I don't want to get rid of any tools that we need to get rid of truly dangerous invasive species, but I think this is more cost effective and is a longer term solution. And we did an experiment in which we actually fed the sterile grass carp to chickens. Becca Haney, a former student of mine and then postdoc, did this work. And they did present with vacuolar lesions, but they just kept eating hydrilla. That's not that hard to make a living as a grass carp, I guess. So I think it's a thing, we need to make sure they're large enough when they're stocked. 10 to 12-inch fish are more likely to survive than the little tiny ones that we tried in the lab. They didn't survive very long at all. And we have done a number of studies in which we incorporate some early herbicide and then use grass carp, so that we're not in a time period in which we have waterfowl on there. We don't have a lot of growth of hydrilla yet. But yeah, the fish end up with the lesions in their optic region as well. And I thought this was a good slide that Al Camus made for me to show how different the fish that have been consuming that hydrilla look. So the thing that's to me most concerning is that this is a lipophilic compound and it's persistent. So I think it presents another toxin that we have to worry about in these environments. And in Florida, all the water is connected. So I think that it is possible that even after the hydrilla and the toxins are gone, that the toxin might still be there in some fishes or in some prey base. So we have a lot more work to do on this. I thought that was a really cool schematic that Tabby made to show the overview. So at this point, we are able to synthesize it. And so that really helps with doing some future work with toxin testing. And we're planning to do the mouse trial, which is sort of the definitive cyanotoxin determinant next month at UGA with the vet school. They have really good people there. And we also now have all the gene clusters that it takes to build the molecule. And Linka Stenklova from the Czech Republic has the PCR methods for that, as we can detect the cyanobacteria with PCR, but we can also detect those gene clusters that produce the toxin. And so we want to develop a LAMP assay, which allows you to go out in the field and use this cute little like Barbie PCR thing that has isothermal so that you can actually use it in the field and see whether or not you have those toxin gene clusters. So I have a lot of questions, but I guess I am going to be funded from Georgia DNR and Pittman Robertson funds because it's affecting game birds and mammals, as far as we know. And we're looking at wood duck nesting success, which seems to be failing, and the lakes where the eagle nests are failing. And I think the prey base may still be toxic in the spring whenever they're nesting. So we're going to follow up on that. I wanted to talk with some more people that are doing contaminant work on that. And are these safe to consume? Honestly, I don't want to go into another hunting season where some of my friends at Warnell are going out fishing and hunting. So, and not be able to tell them, might not be a good idea. So we have some evidence that mammals are susceptible, even without the mouse trial. How am I doing on time? I'm probably talking too fast. I'm from the north. We have worked so much with different folks that live out these reservoirs that are really interested and tell us what's going on. I have this guy that found this lethargic beaver under his dock, would always call me as soon as the coots were coming in, and he'd start watching them and seeing if any of them were sick. So having observers there at the lakes has really been helpful, and working with the reservoir managers rather than we bring our boat, because I don't want to spread stuff around. We go to them and go out with them, and they know what they're doing, and they're also more invested in it. So I think it's been a plan that we will continue to follow. And we did find the cyanobacteria as well as hydrilla in the beaver's gut. You know, they eat a lot of wood, of course, but they will eat a lot of submerged aquatic vegetation if it's available like this. And more recently, we got some slides from Squidus, which confirmed that this beaver did have the bacular myelopathy presenting in the cerebellum and cerebrum. My newest worry are these little cats from Florida. The Florida panther and the bobcat are experiencing strange new disease, feline leukomyelopathy, in Big Cypress National Preserve, but it looks clinically and histologically a lot like bacular myelopathy. And we have found the toxin in the tissues from these bobcats and panthers. It's still ongoing work, but I am concerned that we have our top predator down here in the Everglades with this toxin. I didn't even know it would be down that far. Oh, so we'll watch something cute. These survived. So it seems like the mothers when they're getting sick, they're having kittens that are already debilitated, and this makes sense because it's a lipophilic compound. So the mothers carrying the babies are burning up some of that fat, and then when they're nursing, they're going to be downloading some of that toxin to their babies. So these kittens survived. One of them's still just oversleeping. This little guy looks happier. There's so many things that could be affecting them down in this region. Certainly we know lead and mercury have been a concern for a long time, and there could be synergistic effects between this toxin and those if they're both messing with calcium channels. So I think there's more work to be done there too. Oops. Let's start that over. Sorry, grandson. Okay, yeah. So yeah, this explains a lot, I think, about. Oh, and the other sort of interesting new finding I had this last year, because a student of mine reached down into Lake Oglethorpe, which has no hydrilla, and pulled out pine needles and said, "Oh, you'll like this, Dr. Wilde. Look, it's all nasty and blue-green." And the toxinous was growing on the pine needles, like in the intersection wherein they also have bromide and pine needles. So I think that's not necessarily important in terms of the food web, but in terms of maintaining a toxinous in a reservoir that doesn't even have hydrilla, it would still be there. So, And the water willow that they're planting at Georgia DNR for more habitat, since the grass carp are eating the hydrilla, also supports autochthonous, but it wasn't producing toxin on those plants when we tested it. So we're gonna work on that soon. And yeah, that's what it looks like when it grows on pine needles. Another thing that I've started being more aware of is how valuable the eBird data is. I found large concentrations of herbivorous waterfowl at this little tiny Covington pond here in Georgia. Like birders were going out daily and getting all the great data. And then we had a cold front go through, and then we had a lot of dead birds and feather piles and predator scat. So I don't know how you can tell the difference between like migratory departures and mortality. So that's another question I have for everyone. But I got to meet all of these guys that are doing the work. And I did learn how you're supposed to download the data and evaluate how much sampling intensity there was. So I'm learning a lot at this conference. And I get these updates from Ian Fingston, who's a collaborator of mine on the USGS grant. So whenever we have a new hydrilla infestation, it pops up in my inbox, and then he can contact them and ask for samples. So we're trying to do more of that, especially in areas where it's a new infestation. And we have a map of the probable habitat for, oops, that didn't come out very well, for where we predict based on our positive locations, max scent model. So not taking negatives, but just the positive locations. And it shows up, of course, in the Piedmont where we've seen it before, and then Central Florida all the way down into that Cypress Natural Preserve. And as far out as Texas is where we have this. East Texas, I find it on the hydrilla. West Texas, not. So if anybody's working in Texas, I want to talk to you about that too. And I would love for any of you to send me samples. That's probably the easiest way for me to expand my ability to see it in more places. And it's always good to have controls too. I like to not find it sometimes. And I'm sure that we can provide my contact information to people. And we have the APHIS permits, so we're good. And we certainly have a lot of people to thank for all the work that has been done by a lot of different state agencies and different funding sources over time. And I have a few more. Yeah, there we go. All right, now I would love some questions. That'd be more fun than me talking the whole time.
I've heard of recent die-off, large die-offs of manatees. Have you heard anything like risks for manatees?
Yeah, Florida manatees are probably getting, there's brevetoxin that has been, the Carina brevis blooms have been really bad, and the cyanobacteria blooms, the water column blooms have been terrible. I don't know if they would necessarily, because I have been to several of the springs where the manatees are, where the hydrilla is, and that conductivity is so high, it doesn't, it doesn't seem like that's a good place for the cyanobacteria to flourish, but there's plenty of other toxic cyanobacteria in Florida. I think you have all of the above. Yeah. Yeah, I think the brevetoxin can actually be associated with turtle grass epiphytes as well, like get sort of stuck on it, and then the manatees are consuming it and actually getting it after the fact. Even when Carina dies off, the toxin can still be present for a while within the plants.
Okay.
Oh, sorry, yes.
Have you thought about what the pathway would be for this toxin to get into the Florida Panther?
Yeah.
How would it get in?
Well, those apple snails and raccoons, and now we have these terrible snakes that have eaten all the other small mammals they used to eat. Now they're eating their eggs. So I don't know. I think there's a lot of pathways within that aquatic environment down there. And they noted that these die-offs started in 2017 is when they really started to see some sick animals. I'm like, what happened then? Irma? So I'm a little worried at post Ian that we might spread some more things around because it takes the hydrilla out of some lakes and just throws it, anywhere. So I do feel like that is possible within their food chain. Even deer that will go out and consume some of the hydrilla, they eat plenty of deer. So we're working now with Mike Chamberlain, and he has done a lot of work with the black bear and also done some work with the deer down in that area. So we have lots of cameras to get data on that. That's the next project.
Thank you for really. And my question is, I was wondering about the concentration that they have to eat to be infected. And it probably varies with species potentially, but can they eat it at low levels for a long time? And or do they need, and they like spike that concentration, or would it just build up over?
Now, this is guessing, but probably the larger body size, yes, you could have small doses over time and it might accumulate in your tissues, but you wouldn't necessarily have any problems with it. But if you were, say, to have a baby, then you might be burning up some of that fat reserve or using it a different way, and then it's mobilized. But yeah, I think it can be both like a long-term chronic problem, but acutely, if it's in a high enough dose, it kills the birds within a couple of days with a single dose. So it can be fast, but they're tiny weight compared to a mammal generally. Well, there's not that much bromide in the water. There is bromide is more of a concern, and because we're using it for contamination in water supplies. But if you have a combination of a coal-fired power plant, plus you're using some bromide in your treatment, that tends to make it elevated in the water, but it's even higher in the sediment. And then I guess what I'm saying is that the amount of bromide that was in the hydrilla is much higher. So I think it can usually be, it would be stable and not necessarily a problem if it's in the sediment, but if you pull it up through the plant roots and into something consumable, then it's a problem. Yeah, I don't think we get rid of a bromide entirely.
What are the reaction to state health organizations to your findings?
Well, I have gotten their attention. EPD has been working with me, and the Georgia fisheries guys are collecting fish from all the same sites that they do their fish consumption advisories for mercury, lead, PCBs. So we just need like a small chunk of the flesh of the fish to be able to detect and to be able to analyze it for the toxin. So we're starting to do that, but I haven't, they're not funding us yet on that, but they're sort of helping us with the materials to do it. I think I'll get more attention if we have the mouse trial completed, because that's just sort of the gold standard.
Is there any evidence that most bird or other animal show symptoms that you can reverse it by putting it out of
Yes. So one of the researchers, a veterinarian in North Carolina, actually got coots that were sick from Wood Lake and kept them at NC State, and they resolved as far as like their behavior. Some of them died, 6 of the 30 died. But then the rest of them, once they were just fed, they were behaving more normally. But at the end of that trial, a 30-day trial, they did the necropsies and they had the vacuolar myelinopathy. So I think it's possible that birds that were exposed but survive would still be able to live, but I don't know if they remember how to migrate or things like that. Like the wood ducks leave my AVM sites are going crazy places. So they're not normal migration, doesn't look like. But I'll know more about that next time.
Okay, so we talked about this earlier. You just have like what happens when you try to bring it into the weeds.
Well, this is like a fantastic way to concentrate a bunch of sick waterfowl into locations, and then birds of prey are targeting those situations and eating a lot of. Yeah, I think it could be. There's so many things between infectious diseases and toxins that are interactive that make them more susceptible to that. I don't know why this year we've lost so many vultures and eagles, like 2021 was terrible. In 2022, yeah. So I am concerned about avian influenza, and I think having a toxin in the environment is going to make it worse, and concentrating our waterfowl is going to make it worse.
Osprey population in Florida.
Well, that's what I worry about, like when you stalk a grass carp or the osprey, like going to go for that. I think it would be opening a window of risk for piscivorous predators, but hopefully that outweighs the fact that you've gotten it all out of the lake. But yeah, we did have some sick ospreys at one of the sites, and cormorants that were sick at the first grass carp stalking, but I felt like those were a little small. I think it helps also to stalk them big enough so that they're not a good prey item.
What about pelicans?
I don't know yet. It's a good thought. Bad thought. First question is that wait, ask me the first question again. I already forgot it. So we're working on the second one. Yes. Sorry. So yes, it's not we don't find it in high concentration in the liver, which is what you would expect with many toxins, but we do see it stored in adipose tissue and in the muscle tissue. And then the second question. Brain, we're in the brain. So in the optic lobes for the birds, specifically initially, but then it can be throughout the cerebrum and the cerebellar tracts, sort of as it progresses. But for the snakes and salamanders, we saw it in the olfactory nerves as well as in the cervical spinal cord. So I think it depends on, what that animal needs to survive, like what's the most important nervous system that it hits upon. She's having trouble remembering questions. Go ahead.
How many coops?
Oh, well, the problem with the eagles eating the coots is the coots probably have a belly full of hydrilla also, and they eat everything. Like they eat them down to their feet, leave those in the nest. But it seems to be that because of the progression, it may take a few of them, because we see it in waterfowl in, say, November, and then it's usually a few weeks later before we start to see some factors. But we don't know the dosing level. That's just sort of an estimation. But yeah, I think that's a bad that's a really effective way to transfer the toxins to the top predator. Thank you all very much, and I'll be happy to answer more questions later.